welcome all the participants to the 9th webinar in the series of webinars conducted by the Alliances for the Coconut Industry Development for the Caribbean. This webinar is entitled Coconut Mother Palm Identification and Seednut Selection. Before I proceed with the training session and agenda for this morning, I would like to recognize the European Union, which through the 11th European Development Fund has fully financed the implementation of the project, Alliances for Coconut Industry Development Expansion and Enhanced Support for the Caribbean. We also want to recognize the CARIFORUM Secretariat, which has also made this project possible under the Caribbean Regional Indicative Program. We acknowledge the Coconut Industry Board of Jamaica and the stewardship and collaboration across the region, as evident by the presence today. And last but not least, we want to recognize the International Trade Center, with whom CARDI is co-implementing this project within the identified CARIFORUM countries. Just a few housekeeping matters before we proceed. Let us keep our mics muted during the webinar. Questions are reserved at the end of the presentation by raising your hands or putting your questions in the chat. We would like to conserve bandwidth, so keep your videos turned off and turn them on if you want when you intend to ask a question or you're speaking. All participants should ensure that they're registered and the names used when participating should be the same. I will now hand over to Tristan Alvarez, who's the CARDI project manager and is the Deputy Program Manager for Resource Mobilization, Planning, Monitoring, and Evaluation in CARDI, to give an introduction to the second phase of the project and the webinar. Thank you. Tristan.
Thank you, Dr. Robin, and good morning to, let me say good day because it's perhaps morning and afternoon or evening in other parts of the world. So good day to everyone who has joined us for this webinar, which will be on coconut mother palm identification and seednut selection, a topic of interest for a lot of persons. I too want to echo Dr. Robin's acknowledgement of the European Union, who has made this project possible by financing it through the 11th EDF. I also want to recognize the CARIFORUM Secretariat, who has played a major part under the Caribbean Regional Indicative Program in terms of bringing this to fruition as well. The Coconut Industry Board, their stewardship to the region in terms of development of the coconut industry is well known, and we thank them for being here today as well to continue that work. And our friends in the ITC, the International Trade Center, with whom we are co-implementing this project. And let me just mention that this project is phase 2 and a follow-on of what was phase 1. Phase 1 was a project implemented during the period 2015 to 2018. And it was implemented across very specific CARIFORUM countries. Much of the work has been lauded, and it was through those efforts that we've been able to secure funding from the EU to continue this phase 2, which began in 2019 and will end in 2023. We have also been able to expand the number of countries with whom we are doing work, and we're also grateful for that. Now, we know that the times has now forced us to utilize technology in more innovative ways, and hence this training webinar is being done virtually, whereas we would have been in the field in the various countries. So this platform has even allowed us to share the benefits of our expertise and experience beyond the focus countries of the project. So we are grateful to have this wider capability to reach a wider audience so that we could share the knowledge and expertise and experience of the Coconut Industry Board and other stakeholders who have lots of information to share. Just to put in some context, there was a planning meeting in December of 2018 where a number of stakeholders who I would deem very relevant in terms of expertise, such as the Coconut Industry Board, CAFSA, the University of the West Indies, CICI in Mexico, amongst others. They would have formulated and agreed upon a three-pronged strategy to address the issues surrounding the unavailability or lack of availability of coconut planting material. So these three strategies are as follows. One, the importation of coconut seed nuts from third countries, those external to the CARICOM and CARIFORM region. And while we ponder on that approach, we still recognize that there's a need to import material outside of the region in a very safe manner. And part of the discourse was that of a consultative body which could support such procurement while recognizing that it can't provide guarantees because of the sovereignty of nations when it comes to their borders and plant protection and quarantine officers. However, this body would increase the likelihood of securing planting material which is safe, disease-free, and likely to be entered into the country, and so it would be a service to the region. Now, this is still in the works, but it's something that has been looked at as one of the strategies, which is the importation of coconut seed nuts from third countries external to the CARICOM, CARIFORM region. Second, we're looking at the importation of coconut in vitro or tissue culture material from available sources. For example, CICI, Mexico. Now, while that is one of the strategies, we recognize that work needs to continue within the region across the tissue culture laboratories in terms of achieving the one-to-many multiplication technique with regards to coconut germplasm. Okay, so being able to mirror the work that CICI has been able to have such success with, but the strategy being to import the in vitro tissue culture material from available sources. And lastly, the third strategy is what we're discussing today: coconut mother palm identification and seed nut collection. Basically encouraging the development and strengthening of national programs and having a strengthened national supply base for coconut planting material in the various countries. So this is what today's webinar will focus on and be all about. While in the future we expect to have webinars on the other strategies, we ask your indulgence with regards to this particular strategy, which deals at the national level and really seeks to target those technicians and extension officers who would be in the vanguard in the ministries of agriculture and other technical schools of thought who will be able to lend support in terms of ramping up the supply base within the various CARIFORM countries. So with that said, let me also acknowledge the ACP group of states, the Africa, Caribbean, Pacific group of states. They have also lent much support to the project, especially during phase 1. And let me now hand over to the moderator, Dr. Robin, and I thank you again for the opportunity to share these words and also being able to share the expertise of the team, Coconut Industry Board, CARDI, and the ITC International Trade Center on this webinar. Dr. Robin?
Thank you, Tristan, for the background and the efforts that have been made and the plans for addressing the issues of scarcity of planting material. The next presenter, Dr. Millicent Wallace of the Coconut Industry Board in Jamaica. Dr. Wallace was employed in the Ministry of Agriculture from September 1980 to July 1989, mostly in research and the development division. She graduated from the University of West Indies Mono Campus in 1992 with a BSc Natural Sciences, chemistry major, botany minor. And she received the Don Skeldin Prize for first year botany. So Dr. Wallace has always been an excellent scientist. She graduated from the University of West Indies Mona in 2013 with a PhD in biochemistry. Her thesis, the genetic analysis of coconut populations in Jamaica, featured in 2013 Research Day booklet of the University of the West Indies. Dr. Wallace is presently employed at the Coconut Industry Board, and that is from October 1992, through present as a botanist, plant breeder. She's also been appointed as Research Director from March 2015. Dr. Wallace will present today on mother palm identification and seed nut selection. I will now hand over to Dr. Wallace, who will present on the topic. Dr. Wallace.
Thank you very much, Dr. Robin. Good morning, everyone. Thank you for inviting me to speak, and it is a pleasure and an honor to share my experience and knowledge on the topic of coconut mother palm identification and seed nut selection, gained mostly from my years as plant breeder and botanist here at the Coconut Industry Board. Next slide, please. Just a very, very brief introduction before diving in. The coconut crop is planted over 11 million hectares in over 85 countries worldwide. And approximately 96% of the crop is cultivated by smallholders, which makes it very important. Coconut is indigenous to some countries, but not to others. And that means that the countries to which it is not indigenous, like Jamaica, we had to do our collection from overseas. But the countries that have coconut indigenous can survey their local population for the identification of mother palms. Next slide. The relevant botany of the coconut palm. It may be described as monoecious. Both the male and the female flowers are present on the same palm. Both are present on the same inflorescence, which is emitted approximately once per month. And that is so important because as a smallholders crop, it makes for income that is spread throughout the year. Next slide. When the inflorescence opens, the male flowers open first. They start shedding their pollen first before the female flowers. At some point, the female flowers will start to become receptive to pollen. And what happens is that there are 3 stages. In the pre-receptive stage, the ovary does not open. And then there is a receptive phase where the ovary opens and pollen can be accepted. And then there's a 3rd phase, the post-receptive phase, where the ovary closes and no more pollen is accepted. So the receptive phase is just a short window of opportunity where the plant can be pollinated. Now the male and female phases on each respective inflorescence may or may not overlap. And we're going to look at that a little later on because it influences the collection and ultimately the breeding process. That being said, there are some varieties that even though the male flowers on that variety have all been shed, when the female flowers become receptive, there are few cases in which pollen from the next inflorescence may be able to pollinate those flowers. And the slide you're now looking at shows the coconut inflorescences. The male flowers are at the top of each spikelets. In real life, they look like giant rice grains. The female flowers are further down and they look like tiny buttons, or as the farmer would say, baby coconuts. And those will develop into coconuts if fertilized. Next slide. And this is a close-up now of the mature male flowers. Can you see the pointer, anyone? Can you see my mouse? Yes, no?
No, I'll take
Oh,
responsibility.
all right. In the right-hand corner of the mature male flowers pictures, you can see an open male flower. But the others, as you can see, look like giant rice grains. Right hand and bottom, those are open. So what you'll find is that the flowers will open from the top down, and as soon as they shed their pollen, they fall off. On the left-hand side of the slide, the receptive female flower. Looking at the middle, there's a white section that the tripartite, the teeth of the ovary, and that opens, and that tells you that the coconut will now receive pollen. Also, a drop of nectar is produced there, and that is why the ants are attracted, and also bees and other insects. They come to enjoy the nectar, but at the same time, they leave pollen behind because they are going from flower to flower, and this assists the pollination process. After when the coconut becomes post-receptive, this white area closes off and becomes brown. And you can see that from the ground. Okay, they're post-receptive. Next slide. There are three main types of coconuts. There's a tall type, high height increment. It means that it grows at a faster rate. So a tall, a dwarf, and other types of the same age, if you place them together, the tall will look a lot taller because it grows faster. And you can see from the space leaf scars, and it's predominantly allogamous. I'll speak a little more about that in the next slide. And it's late bearing. Talls tend to bear like 6 years or more after planting. And then there's a dwarf, it's shorter, more narrow spacing between the leaf scars, predominantly orthogamous, and precocious means it's early bearing. And there are a few intermediate and other types, like the Nulika dwarf. It is referred to as a compact dwarf variety. And the king coconut, which is an intermediate it looks between the dwarf and the tall, but it's not a hybrid. And of course, there are hybrids, which are genetic crosses between individuals from any of the above types. And those will display the characteristic of both parental types. Now, when you're going to collect or to identify a mother palm, this is one of the things that you need to be able to determine. Is it a tall variety? Is it a dwarf? Is it an intermediate, or is it a hybrid? Next slide. Allogamous means the falling of male flowers before female flowers become receptive. No overlap of the male and female phases. So if it's going to be pollinated, it is most likely by pollen from another palm of the same or other variety, or in a very small number of cases, by the pollen from the next inflorescence. So we refer to talls as outcrossing, and that will bear heavily on the breeding process. Orthogamous means the female flowers are present. Male flowers are still shedding pollen when female flowers become receptive, meaning that an overlap of male and female phases, and it means that pollination is most likely from the same palm. It's self-pollinating, and that makes it easier to breed. Next slide. And these are pictures showing you the tall varieties. And the tall variety I've featured there is the Thailand tall from my collection, the board's collection. The dwarf variety featured is the Sri Lanka yellow dwarf. Next slide. The intermediate is the king coconut from our collection. And of course, the hybrid is our maypan. And okay, looking at the maypan, you are able to see that at the base of the trunk, there's a little swelling. We call it a bowl. Right. It gets that feature from the tall parent. For the intermediate, the king, there's just a little bowl. And if you could go back to the slide before, the dwarf, most dwarfs do not have a bowl. And that will influence the windstorm resistance of the plant of that particular variety. You find that the taller varieties are more resistant to windstorm because of that bowl. They sway, more likely to be swaying in the windstorm than to be toppled over, which happens a lot with dwarf varieties. Can you forward, please? Next slide. So in identifying your mother palms, you are going to need to survey your intended populations. So you're going to visit and you're going to do some homework. You're going to examine the history of the palm. Do they have a good performance record? And some of this information you will be able to glean from local farmers, because if they have been growing these coconuts, they will be able to tell you something about it. Do they have a good performance record? If you're going to have a mother palm, if you're going to do breeding, you need to start with good material, good traits that your offspring will have good traits as well. And then you're going to ask if there are local land races, if the coconut is indigenous to your country. Is it a local land race or is it an introduced variety? And of course, do you know the name or the parochial name of this variety? And then you need to find out if it's a hybrid or if it's a land race that has been cross-pollinated and become a hybrid, an unintentional hybrid. That will affect your offspring. And of course, this information that you will need to get it from persons who live in the area or work with these coconuts. But you need to know the history of the palm that you're looking at, your candidate palm. Next slide. And then, of course, you need to examine the environment in which a palm is situated. Do these palms appear to be surrounded by other palms with similar trait? It makes life a lot easier because we call this uniform blocks. And you'll hear me refer to it a couple more times in the presentation. Are they in uniform blocks? Or are they surrounded by other varieties that, obviously other varieties are mixed stand, and that is going to make life a little bit more complicated. And then of course, are these palms easily accessible? Are they on reasonable terrain? Are they on farmers' field or properties that you can access? Are they too tall to be reaped or pollinated if necessary? So you have to examine the environment. Is the environment suitable or agreeable to my making this a mother palm, doing collections or otherwise? Next slide. So now you're going to examine suitable candidates. There are some desired traits that you would like. So you're going to look for those traits in the mother palm and of course the pollen parent if not present. You're going to look for genetic integrity. In other words, is it a true to type variety? Because if it is not, then your offspring will not be true to type. And the nuts produced may be very good for farmers, very good for processors, but for breeding, a certain amount of genetic purity or genetic integrity is necessary if you're going to hope to have offspring of the same type with the same desired traits. Then you're going to look for resistance or tolerant to important pests and diseases in as much as possible, because as one researcher puts it, the best coconut, the best bearing coconut is no use unless it is alive or in good serviceable condition. If it's dead or very damaged or very impaired, then it's not going to be of much use to you. So it needs to be standing up to the stresses, biotic and abiotic, that it will face. Is it performing well in that area? Then of course you're going to look for high stable yields. The farmer will want high yielding coconuts, and the yields need to be stable, not a lot during 1 year, and if there's a very, very dry period, then little or none. There will be differences, but the yield needs to be more or less stable. And the farmer will thank you for that because he wants to know that he can count on the yield because that is where his profit is going to be from. Next slide. So a little more explanation of genetic integrity. The preservation of advantageous traits. If the mother palm, let's say I'm Jamaican, good resistant to lethal yellowing, and I have now made offspring that did not preserve that trait, I will find that the offspring don't survive as well as the mother palm, and that will be a problem. So we need to make sure that the advantageous traits are preserved. For an original cultivar, that's easy as long as you don't hybridize. You keep the cultivar pure. And we're going to look at ways in which we can do that. In the case of a hybrid, like the Maypans, of course those are bred in. Because for the dwarf by tall hybrid, like the Maypan, the Malayan dwarf has a high female flower production, which means that it will bear many nuts. And then, but it's small, but nuts are small. So we look to the tall, like the Panama tall, with large nut size. So in making that hybrid, we try to get the best of both worlds. Many nuts, good size. So it will be bred in for the hybrid. For the original cultivar, make sure you preserve it. Next slide. For resistance tolerance. Of course, in this region, it has to stand up in some measure to lethal yellowing disease. We had a very good cultivar called the Jamaica Tall. Very good oil content, very good copra, really very nice. But sadly, it was very, very susceptible to lethal yellowing disease. Now there's hardly a Jamaica Tall palm around. And it was sad to see that cultivar go because it had so many things, good things going for it, except resistance to lethal yellowing. And even though all the cultivars have been affected, you will find that some have slower mortality rates than others. And of course, you have to look at other diseases of importance like bud rot and other diseases in your particular region, because your mother palm of choice needs to stand up and needs to pass these features down where it stands up to the stresses that are present in your region. So resistance tolerance is a very big consideration. Next slide. High stable yields. Well-performing local land races are often desired. And of course, it's going to be a heavy influence in your choice of mother palm, because the more coconuts produced, the better the yield, the happier the farmer will be. Next slide. So having selected your candidate populations, now you're going to evaluate. And the best way to do this is to collect some preliminary data. The farmer will say to you, it bears very well, and it does bear very well, but you need to put some numbers to it. How many nuts per bunch? How many nuts per annum? So you're going to need to collect that sort of data to begin with, that you have numbers to speak about, not just it's very good. Large nuts, of course, you need to measure to say the polar diameter is this, the equatorial circumference is this, and the volume is this. So you can communicate to someone to impress them of the beauty of this hybrid, this variety. And you need to do this by sampling random palms. If you have a large population, if your population is small, you may be able to sample all the plants. So some of the important characteristics that you would measure in this preliminary data would be the yield, of course. And the easiest way to do that is to count the number of bunches 5 months old and up, because it is believed that even though the fruit is set by 5 months, those that will fall, and those that remain will be carried to maturity. So we really start counting at bunch 5. And the easiest way to do this is to count all the bunches to maturity. Say they're 5 to 12, that would be about 7 bunches. And then you count all the nuts in all the bunches, and you make a simple division, and that would be mean number of nuts per bunch. Of course, you take a mean for all the palms, but that will be nuts per bunch, and you get a good estimate of your yield. And then you need to look at the stem and leaf morphology. How much does it measure at the base? How much does the stem measure? And it will help to tell you the type of plant you're dealing with. How does it perform in response to diseases, dry condition, windstorm? What is the survival rate? Did a hurricane pass through 5 years ago? How well did they do? And of course, ease of collection, reaping, pollination. And I pause here to make a comment. I started doing the hybrids in the 90s with promising varieties from India and Sri Lanka, and I crossed each one with the Panama tall and got a set of hybrids. Now I use the Chowgat green dwarf, Sri Lanka green dwarf, Sri Lanka yellow dwarf, and the king coconut. But I had so much difficulty getting the king coconut hybridized. I got about half a dozen seedlings for my troubles, and I had to drop this one from that particular breeding program. And I also found out that the new Lika, as impressive as it looks, that's a Fiji dwarf, could not be used very extensively in the breeding program because the compact nature of the crown made pollination and other matters very difficult. So you need to look at that. How easy is it to collect and reap and pollinate? And there's another characteristic I didn't place here. I'll speak about it later. It's the 11 scar length. Now you measure the stem to 1.5 meters from the ground, that's about chest height, and then you count the next 11 leaf scars, and that will tell you the distance that the trunk grows after 10 leaves are emitted. And you will find that for the tall variety, it's a lot greater than the dwarf, and the intermediate and the tall dwarf by tall hybrid would be in between. So with experience, you can enter a field and look at the leaf scar, the 10, 11 scar length, and make a judgment as to whether this is a tall or dwarf variety quite easily. Next slide. And yes, I was ahead of myself, but that's fine. Is it a tall, dwarf, intermediate, or hybrid? You need to figure out, because it will affect how you collect. And of course, if floral biology is going to give you a good indication, look at the flowers that are open, inflorescences that are open. Do you see male flowers still? Are the female flowers beginning to become receptive? If they are, do you still have male flowers present, or are they, have they fallen already? And this will tell you, give you good indication whether it's a tall, a dwarf, or a hybrid, which is in between. And as we are going to see, it will inform the choice of your collection strategy. Next slide. You're looking for overlapping of male and female phases. When if male flowers are present, when female flowers become receptive. Go ahead, next slide. So now you're going to mark your palms of interest. There's no point collecting from a palm, receiving some good material which you'd like to repeat, and you're not sure where you got it. So you need to now develop a system. Say you found a population and say, okay, these are good candidates to be the mother palm. You need to develop a system of identification if it's not already present. If you know the variety, you can have a symbol for that, and you can number the palms 1, 2, 3, 4, 5, 6, or measure in rows, row 1, palm 1, row 2, palm 1, 2, 3, 4. But you need to have that a unique identifier for each palm. Like when we had like the king coconut population, we had K1, K2, K3, different from the Shawgat green, which would be C1, C2, C3. And you're going to attach that identifier to each palm. You can paint it with paint and a brush or spray paint, or you can paint it on a metal tag like an aluminum tag and nail it to the trunk of the palm, or you can make a sign, and if the palm is very young, and stick it in the ground beside the palm. So later on, if you get desirable progeny, you can always come back to know which palm it came from. Next slide. So it is a very good recommendation to collect and set a preliminary sample of seed nuts. And then you set them in the nursery, look at the seedlings that are produced, and it will give you valuable information on the genetic purity of the mother palm. If it is a hybrid, like the Maypan, you're going to get a proportion of seedlings that I'll speak about later. If it is not pure, you're gonna see it in the seedlings in the nursery. And something that you mainly look at is the color of the petiole, the leaf, the color of the petiole of a leaf of the mother palm very likely correspond to the color of the petiole of the seedling in the nursery. And you're going to use that as a marker to say whether what I'm getting here is pure. It could be that the mother palm is okay, but the contamination happened in the pollination process, and there are ways to deal with that. Next slide. So the first thing you can do is to reap the existing seed nuts from the palm. These would be, of course, open pollinated, male parent not known. And this is the most straightforward and easiest method of collection. And over the years, there's a way to do it to get good material. Is if you have a uniform block, like I said before, if you have a collection of one particular variety, no neighbors, little or no neighbors of another type, you just go into the middle and you collect the seed nuts from those, because those at the end might be influenced by pollen from elsewhere, but those in the middle, there's a very good chance that they're getting their own pollen or pollen from their neighbors. And if their neighbors are the same, then there's a good chance that you have good material. And of course, you're going to label your seed nut, the same identifier that you put on the palm. You're going to label your seed nuts with the same number, the same identifier. And if you're doing a lot of collection, you can save yourself a lot of trouble by, when you collect all the nuts from one palm, you place them in a bag and you label that bag. Or what I see them do in West Africa, because they really go palm to palm, they use a large upholstery needle and twine, and they sew the husks of all the palms from all the nuts from one palm together. So here's a bundle. All of that came from one palm. So when you're done, you don't have to go sifting through the heap of reap nuts to find the ones from the different palms, because they'll be all together. So that is going to be very important. Next slide. So if you're going to plant the open pollinated seed nuts, you need to know this. Those that you would have reaped the existing nuts. If it is from a tall variety, there will be most likely genetic contamination, because tall varieties take pollen, are most likely pollinated by their neighbors. This, of course, is mitigated by collecting from the middle of a uniform block. If the seed nut is a dwarf variety, self-pollination is more likely. So you'll be getting away with good material, even if you just pick the open pollinated nuts. Next slide. Of course, if you plant the seed nuts from a hybrid palm, and you're gonna see this, start seeing it in the nursery, and you will see it later in the field if you do, the segregation of the genes will occur. So half of the offspring will be hybrid, a quarter will look like the pollen parent, and a quarter will look like the mother palm. And we saw this very much in one of our land races we had. The Fiji Malayan orange dwarf, it came to us some decades ago. But what it is a new Lika cross with the Malayan tall. So we planted them and they're nice. But when we collected offspring from those plants and planted them the next generation at another site, it was plain to see some of them look like the mothers, some were short, just looking just like the new Lika, and some look like the Malayan tall. So if you plant seed nuts from a hybrid palms, that is what you're going to get. You're going to get a non-uniform population. But like I said, you will begin to see this in the nursery, especially if the parents of the hybrid have different colors. You will see this coming up, because in the nursery you'll see the difference in the petiole color. So you need to know if what you're looking at is a hybrid. It is not the best candidate for a mother palm. Next slide. If all this fails, then you might have to resort to controlled pollination to preserve the genetic purity of the desired varieties, especially those that are in mixed stands, because sometimes you just can't help it. You might have a good variety, but it has other types around it, but it's promising. Like I say, if it's a uniform block, no problem. For dwarf variety, it's simpler because you make a bag of a certain, dimensions enough to cover the entire inflorescence. It would be like a tube wide enough to fit over the inflorescence, open at both ends. So you're gonna slip it over the inflorescence and tie both ends. You wait until it's open, the inflorescence comes out and pollen start to be shed, and you're gonna put that bag on, and you're gonna keep it on for a couple of weeks. And after that, you check to see if the female flowers have become post-receptive. Then you can remove the bag, and you have self-pollinated nuts. For tall varieties, it's a little bit more complicated because it would mean that you can do sib pollination, where you collect the pollen from all the individuals and you pull that pollen and you emasculate. You will have emasculated, collected the pollen, processed, pull all the pollen together, and then you apply them to all the female flowers. So it will be pollinated by its sibling. It's a lot less complicated than doing those for each tree. Only some research stations do that, but we do sib pollination when we are replicating our tall varieties. Next slide. I pray that you get a good mix, a good uniform block. So now you're setting the seed nuts in the nursery. You're gonna set the seed nuts with the same identifier together in a bed, a bed all by themselves, and you're gonna make a sign for the bed saying these are seed nuts from this particular palm, and of course the date that they were collected and the number collected. And of course the seedlings that you're going to get, as soon as they grow, you are going to need to tag these seedlings with the same information, that all through the process you know what you're dealing with, and when you're ready to plant out in the field, you knew exactly what it is, where it came from. If it did very well, you know where to go back to get more of this particular one, and you will know the ones that you need to go forward with. And in that way now you will have begun the process of identifying your mother palms and selecting your seed nuts. Next slide. And this is where my presentation ends. I am not a lady of many words, so I think I may have finished in a shorter time, but I'm willing to answer any questions that you may have based on my experience. Thank you very much.
Thank you, Dr. Wallace, for a very precise but detailed presentation on mother palm selection, detailing the morphological characteristics and the insights into the use of pollination to get the type of characteristics you want to get.
Right.
The floor is now open for questions by show of hands. I see one question in the chat from Raymond Trotz, my friend from Guyana. I am going to read out the question. Before we continue with the question and answer sessions, I want to say that Dr. Wallace is supported by Dr. Wayne Myrie from the Coconut Industry Board and Dion Clark Harris, the entomologist from CARDI, to answer your questions. So you have a tripartite arrangement. Dr. Wallace, Dr. Myrie, and Dion Clark Harris from CARDI will be answering your questions. I will read out the question of Raymond Trotz. Can we have a regional molecular characterization program for uniformity of identification? I think I understand that part clearly. And then he continues, an ease of husbandry through cross-reference in pests and disease and best practices. The second half, I'm not quite sure I'm clear on that question on the second part. But Dr. Wallace, Dr. Myrie, and Dion, that's the first question I see in the chart.
Well, I start the ball rolling and hand over to Dr. Myrie. For my postgraduate research, I looked at the populations and I used two molecular methods, RAPD, that's rapid. I can explain if you like, and AFLP. I did get differences with the RAPD, but not with the AFLP because they were the same species. I know that Dr. Myrie has done some work with microsatellites, which was better able to do more resolution of the varieties. But it's just that the coconuts, because one species, not so easy to identify on a molecular basis. Nevertheless, that kind of work still continues. Dr. Myrie?
Thank you very much, Dr. Wallace. Good morning to everyone. Yes, you are correct to say, Dr. Wallace, that the microsatellite techniques can be used. It gives further resolution, of course. It is a better technique to use than the rapid random amplified DNA polymorphism. It is a step up also from AFLPs.
Yeah, AFLP doesn't work, no.
Yes.
Yeah.
So it is a very, and you're correctly, in addition, the coconut variety is a very difficult variety to classify on the molecular level because it is so heterozygous. There are so many genes that it poses some challenges. However, there is a well-known database developed by CIRAD, and the microsatellite technique relies on that database. And we were in the earlier, this is published information, was published by us. We actually look at the Malayan dwarfs, and we did classify Malayan dwarfs here in Jamaica on that basis. And that is how we have the distinction between some of the plants that we have here. So it is quite possible, but it's something that will take a lot of resources so to do.
The second half of the question, I don't think it's very clear. I know Raymond is on. Raymond, the second part of the question, where you refer to the ease of husbandry to cross-reference in pest and disease and best practices. Could you elaborate on that so that the facilitators could address your concerns? I'm not hearing from Raymond, so I think I'm going to move on to another question in the chart. Is there any recent Caribbean experience with Seednuts and plant performance from Cohibra and Technococo in Brazil? Let me repeat the question. Is there any recent Caribbean experiences with Seednuts and plant performance from Cohibra and Technococo in Brazil?
I'm assuming that the two names you called are places in Brazil.
Well, they sound like institutions that use coconuts.
Okay.
Right. I would wish that the participants who ask those questions, if they could add some clarity to the questions, because sometimes the way the questions are written is not quite clear. So that question comes from Adrian Curtin. So Adrian, I do not know if you wish to elaborate a little on that question for the facilitators.
Someone raise a hand? Well, in the go ahead.
Or it may be that the questions were put in the chat before, and they don't
maybe Dr. Robin.
I would like to speak on that. I would like to speak on that. Yes.
Okay, all right, go ahead, Dr.
Or as suppliers, I don't think we have had from those two suppliers, but in the year 2000, we did get a consignment from Brazil, and they have performed very well. And what we have done, we planted them at Spring Garden in Portland, and then the F1 generation, we planted at our main seed garden in Barton Isles. And in 2007, they were introduced into the breeding program, and they were hybridized with the Panama tall. So it would be the Brazilian green dwarf crossed with the Panama tall, we call the Braypan. And we also crossed it with made a limited number of individuals crossed with the Malayan dwarf, and that hybrid would be the Maybraz. And these are being evaluated in a trial presently. And suffice it to say that the Braypan, has done very well, has performed in comparison to the Maypan. The Maybraz, now is a dwarf by dwarf hybrid, gave very good, stable yields. Now, the Brazilians that came to Jamaica were very, very heavy bearers. And we literally had to support the bunches with ropes that they didn't fall out. And when they were hybridized with the Malayan dwarf, those bunches were a little more stable, heavy bearers. And like I say, the Braypan has been released on a semi-commercial basis, and it is now being tried in the farmers' fields. So we did also get some other hybrids from Brazil, but those did not do very well. But I don't recognize the two names of the institution that you asked about.
Okay.
And then also in 2009, we also got some Brazil green dwarf again from the gene bank in Ivory Coast. And those are doing well, yes.
I think the participant who asked the question indicated that these two institutions are suppliers of Seednuts. So
okay.
that could be very interesting for the team.
Yes.
CIB and ITC, that Cohibra and Technococo are actually suppliers of Seednuts, and we may want to follow up on that. Okay. I have some hands up, and I'm going to go. I'm not sure in what order the hands were placed up, but I'm going to go first to Raymond Trotz. And Raymond, you may want to add some clarity to your question, the second half of your question. So go right ahead.
Okay. The question actually is to understand the link between the genotype and the phenotype. For instance, when you look at literature, we see a very large number of varieties from the APCC countries, for instance. And we also have our own varieties here locally, which we call by different names. It's simply a matter of understanding what correlates with what in those countries. For instance, there is the 18-month variety, the very popular 18-month variety. If we can get a molecular characterization of that, then we can understand what it correlates with in, say, Sri Lanka or Thailand, India, etc. And secondly, If we have an understanding of the genotype, then we can understand its susceptibilities to pests and diseases and how it responds to environmental conditions or any changes, any amelioration in soil and so on. So it is from that point of view that I was linking the molecular characterization with husbandry practices. I don't know if that makes it a little clearer.
Yes, understood, understood. Like I said, this work is still in progress, but failing that, what we did when we registered our accessions with the International Gene Bank, we had to send passport and characterization data and pictures, so it could give some amount, failing the molecular information, it could give some amount of explanation as to what you're dealing with, because sometimes a particular variety may have been introduced from another country, and looking at it, they may be able to say, this looks like this particular variety. But the molecular work is still in progress.
Thank you, Dr. Wallace. I will now go to Umaira Belize.
Morning, everybody.
Good morning.
Nice presentation. I really enjoy it. My question is, what is your experience once we identify the mother plants? We have a diversity, so everybody would like to mass multiply this plant. What is the recommendation you give for diversity? Should we continue with Maypan alone? Should we take these mother plants and start to create that diversification on the materials that we have in the field? That is, I think, the question I want to know. When we identify these plants, how will we actually go ahead and make them crops that could be sustainable? Because here in Belize, they use a Maypan and another one called Chatomal, the Brazilian green, and the dwarf. Those are the only ones. Outside, and we identify our mother plants, we will create a diversity, but we also will create a small pools. So I'm trying to figure out what is your recommendation? What should we do? How, as a technical person, should we go ahead and tell the farmers how to go forward with this program? Thank you.
Okay. Planting one variety alone of any crop is always dangerous, because there might be a disease or pest that comes in to which it is very susceptible, and you stand to lose everything. There's an agricultural principle of mixed cultivar cropping, which means that it is safe to use more than one cultivar. But you don't just all mix them together, you plant them in blocks. Maybe an acre or two, you plant them in blocks. Also, if you have a hybrid and a dwarf and maybe a tall, planting them in blocks also works, because the planting distance is going to vary. So my recommendation is as many as you can plant, but plant them in blocks of 1 or 2 acres each, because diversity means protection. Definitely means protection. So you plant a block of your Brazil dwarf, a block of the Maypan, a block of your others, you know. So we always encourage farmers, never plant one variety alone. It is dangerous. If you can manage, if you can help it.
The next, Troy Augustine, I see you have your hands up. Can you ask your question now?
Okay. Good morning, everybody.
Good morning.
I have not heard you spoke about the layout of the germplasm plot. With our recent trip to Mexico City, we were told that if you're doing coconut like a germplasm plot, you should have a buffer zone of around 7 rows of that variety, and inside of that you can have a pure stand plot. Now, I'm saying that because Grenada is not as big as Jamaica or Belize, where you can have large expanse of coconuts. And even with our germplasm plots, we might have challenges because we have a lot of bee farmers, beekeepers, and insect pollination that will take place. So I want to know what is your recommendation when it comes to a germplasm layout plot. And you mentioned that the plants that was imported to Jamaica, in what form? Was it tissue culture? Was it seed nuts? And if it is seed nuts, what is the best means of bringing in the plants? And what are the risks involved? Because from Ivory Coast to Jamaica, it might take weeks or months, and you might, if it's seed nuts, you might have to ship it in a container. That container might have to be refrigerated. I'm not sure. So I just trying to understand what method would you best recommend, and what method did you use, and if there's any risk involved in that venture.
All right, let me answer the second part first. We introduced many seed nuts from Ivory Coast. First of all, they go all around the world, even to Amsterdam. And the risk is that if the coconut goes beyond, is exposed to temperature below 4 degrees Celsius, it will kill the growing point and it will not germinate.
Okay.
So we had to ask for them to be brought in refrigerated containers with the temperature range of 13 to 18 degrees Celsius. Now we lost quite a bit of our dwarfs in the importation process because of this reason. I'm going to explain. And if I'm taking material from Ivory Coast again, this is what I do. They have a tendency to dry their nuts before they plant. So they dried the nuts, then they put them in a container, and they stayed in that container for 6 weeks. So by the time they got to Jamaica, the dwarf, the smaller nuts were dried out, and the germination was a lot lower than we desired. But the tall varieties did just fine. So if I were you, I would ask them to reap the coconuts with living color on the calyx. Don't dry them out. You need your refrigerated container, 13 to 18 degrees Celsius, and that would help a lot. Can you remind me of the first part now of your question?
The layout of the germplasm plot, as we learned from Mexico in February.
Yes.
That the germplasm plot should have a buffer of at least 7 rows in block of those coconuts to ensure that you have a pure stand plot. Because if you take into consideration islands of the Caribbean with small terrain, small holdings, and you want to do coconuts, the neighbor might have another variety that's there and it's not pure to what you're looking for. So what would you recommend in instances like that?
Let me ask you a question first. This plot, are you planning to pollinate? Are you planning to do hybrids? Are you planning to do hybrids or just to collect? The plot that you're speaking about, are you planning to make a hybrid or to just collect seed nuts?
Planning to collect seed nuts, but looking at the land space, it would be unavoidably having coconut that will be crossing because of the insect pollination. Remember, things like insects like bees have a 2-mile, 3-mile radius. So we are not gonna have that. I know that the hand pollination is much different, but the technique needs to be developed.
If you're going to make a hybrid, we would recommend isolation of the entire plot.
Yeah.
If you're going to just collect, once your plot is uniform, then you just have to collect from the center of the plot. When we do trials, we do 1 or 2 guard rows. But if that is your reality, you just work with that reality. You just go to the middle of the plot and do your collection from there. You might not have the luxury of 7 rows, which we don't. Alternatively, if it's dwarfs, you can go ahead and do some bagging.
Last question.
Yes.
does the Brazilian hybrid, the green dwarf, do you have planting material that you can ship to other countries, or it is recommended that we import it from maybe other sources like in Mexico or in Brazil directly?
You're speaking of the Brazilian dwarf or the
The
hybrid?
Brazilian dwarf, the Brazilian, well, basically the varieties that
Oh,
you
the
see.
Brazilian dwarf. Okay, we are just in the process of bulking up our population, so we would not have enough. But you
Okay.
may source them from Brazil. But like I say, if they're gonna bring them, make sure they don't dry out.
Okay.
They're not dried out.
So it's not a problem to bring in seed nuts because there's a little bit of back and forth with should you import seed nuts and the risk involved, and there are persons with influences and has different opinions. So while the project is running and there's a need, only 4 years ago, Grenada got some coconuts from Mexico, the Michoacán, Colema, and a couple other varieties.
Yes.
And one of the plots already started producing. But because of other issues, I'm not seeing that in other farms. Now, there's a hype in Grenada to get coconuts and to revamp the coconut production because we're still importing from St. Vincent, sad enough. And the minister is very interested in turning that around in 4 years' time, 5 years' time.
Yes, yes.
And we need to get coconuts and large quantities, but the roadblocks and the bottlenecks are you bring it in, you might have a risk. You don't bring it in and you bring in the tissue culture plants, we suffered a lot with the tissue culture. Was experimenting and training, but I think there's a more guarantee in having seed nuts imported. And it should be cheaper because the tissue culture plant is $8 for one. Basically from Mexico, $5 at least for seed. So I'm trying to get around all of that and ensure that for the next year planting season, we have sufficient coconuts to give farmers to establish a plot and not just like an acre worth of crops. So you could go in and do analysis on the different areas.
Yes.
And encourage more persons to do it. By then, the coconut water industry, which is very big in Grenada, is gonna see an explosion because farmers are harvesting the coconuts green. You can't get enough planting material. We imported coconut in 1977 from St. Lucia. Before that, we have not had coconuts being imported or after that, except 2016. Now, our problem is that I'm driving through on the sea coast, on the areas where coconuts have been established as over 50 years. The plots are now depleted. They're just standing there with a few leaves and some baby coconuts, and you're not getting a yield. You have trees, but no yield. No, and all of this, a complete replanting drive needs to take place. So I need to get up with the coconut, ensure that we have quantity and quality. So I just wanted to find out what is the best option, and you gave me the option, so I will take that to the minister and the PS and the head of the department to ensure that for next year and in a budget for next year as well, we can get a large quantity from certified and registered plots and ensure that we have the quantity that we need. So thank you so much.
I can make a recommendation. You need to do a little homework.
Thank you.
Find out. You need to find out from the country you're getting the coconut from, what are the important pests and diseases there. If possible, maybe a visit. But I know that from experience, the coconuts that we have imported from West Africa needed to be accompanied by a phytosanitary certificate to say that they are pest and disease free. But when in doubt, a visit may be a very good investment.
No, I was trying to source some from Mexico because of the relationship that we had with CC, and I'm in constant contact with Dr. Opeza and Americ Sid. I think we can maybe get a couple of varieties from them and establish a plot. So thank you so much.
Welcome.
This is a very interesting area of the project, not importation and exportation. And I am fully aware that the project intends to address this. not only at a country level, but at a regional level. And that is going to be the subject of a separate webinar. Where CARDI, the ITC, CC, probably Embrapa, some of the Brazilian agencies, and CARDI
Yes.
will be involved in a discussion, because I think they need, the countries in the Caribbean need guidance from us in terms of the processes needed by CARSA, the Caribbean Plant Health Directors Group, which is made up of the quarantine officers across the region, to develop a system that is sanctioned by us and CARSA to import Seednuts into the country. This is a long discussion we've been having, and we need to bring it to fruition because the countries need the material. So that particular subject will end here. I will now go to the chat to get back on track with the subject matter we're dealing with today. And there's one question from the chat. In seed nut selection, is thickness of husk, mesocap, of any relevance?
Yes, there can be, especially with a tall variety, because what you'll find if the husk is too thin and the nuts fall from the tree, if you pick them by falling, especially the tender nuts, they're going to be splitting open. So what you can do when you're doing your preliminary investigation and you're doing your initial sampling, you're going to cut the nuts open and see the husk thickness. Usually use a saw, and then you're going to cut from one set from the stem end to the flower end, and then you're going to cut some across the equator. And that is going to give you a very good idea of the relative thickness of the husk. And some material that I will share from Cogent, whenever they speak of a variety, there is a board that the nuts are placed on. The whole nuts, large, medium, small, and then you're gonna saw, so you're gonna get the polar section and the equatorial section. And then you will see for yourself how thin or how thick the husk is. But yes, a thin husk, if the nuts are allowed to fall from the tree, they are going to be damaged. It's a matter of your preference.
All right, then there's another comment/question. Harvesting is a problem with coconuts, especially for water.
Yes.
Therefore, should we look at finding varieties that are easier to harvest?
I think that could be one consideration, of course. Another would be the quantity and quality of the water, but yes, easier to harvest. What we do in Jamaica, we don't allow them to fall because it shortens the shelf life of the coconut water. So we actually use a rope and pulley system to lower the bunches from the trees.
All right, there's another question here. Shuin Joseph, good morning. Can you please give some more details in regards to seed nut collection? For instance, what's the best stage for harvesting for seeds? Best conditions for germinating in nurseries, identification, and also the identification of non-viable seeds. So let me just repeat that.
Yes.
Can you please give some more details in regards to seed collection? For instance, what is the best stage for harvesting seeds? That's one question.
Yes, right.
The best conditions for germination in nursery, that's another thing to do with nursery management.
Yes.
And then the other question is the identification of non-viable seeds.
Okay.
So he's asking for the best stage.
Oh, yes.
Harvesting the seeds, and then
yes,
the identification of non-viable seeds.
Okay.
All right.
What we do, our rule of thumb is to harvest at 12 months old. So the nut is dry. but there's living color still on the calyx. We don't allow them to dry too much because, you know, the coconut water can be less. So we harvest with living color on the calyx. When we're setting them in the nursery, each nut, the nursery worker shakes the nut to listen if there's water slushing inside. The only other thing to do would be to cut it open to see if there's an embryo, and then you can't set it. So we shake the nuts to see if there's water slushing inside. The part three, can you repeat part three?
Part 3 was identification of non-viable seeds.
And the part before that?
Okay, that's best conditions for germination in nursery. This is what to do with nursery management.
Oh yeah,
Best
right. We find that if we set the nuts and have covering material, and then we do irrigation and some amount of shading, it will enhance not just germination, but it prevent dieback after germination, especially in very dry conditions. Some countries will do the polybag nursery, but that is really to hold the seedlings for extended period. If you can't do it, you set and cover, irrigate and pollinate, and then you can take it from there. Sorry, irrigate and shade, sorry, yes.
I don't know if Wayne or Dion has anything to add to the...
Please, yes, please, yes.
Well,
Dr. Mary, Ms. Harris?
Dion, I don't know if you have anything to add to the nursery part. Go ahead, Dr. Mary.
Yeah, just to say that coconut is susceptible to desiccation. So irrigation is important, but also covering material of the nuts. It should be covered by an inch of whatever covering material, preferably a very loose covering material so as not to impede germination. And that covering material should not be compacted so that when the nuts are being removed from the nursery, they are not easily, the shoots can be easily damaged. So if you have a compacted material, it will be difficult for the seedlings to be removed, although you're using a spade. So you try to use as loose the material as possible. When you are actually lifting the seedlings, you don't damage them.
Thank you.
And your covering material should be able to retain water, retain moisture. It's able to retain moisture.
I'm going to take one more question.
Hello?
And then I'm going to, oh, sorry, Dion, go ahead. Sorry.
I'm sorry, I was having difficulty unmuting, so.
Okay, go ahead.
My problem. Just to add to all that was said, to support it, but to add that under the project, we have done some work on just looking at all the different ways that have been used in nursery production of seedlings. And we have been looking at orientation because you use different types of orientation as well. And so we have done a trial looking at the diagonal orientation, putting the embryo at 45-degree angle, one vertical, one horizontal, because all of these have been used by different nurseries across the region. And we have found that the diagonal orientation seemed to do better. We are repeating that study. Under the production manual that we used in the project, we also recommend submerging the nuts within the soil a little bit as well. And that has proven to give some help in maintaining the moisture, along with irrigation and shading. So those are some of the things that we have experienced as we have worked with nurseries across the region. We will, in the second phase, be continuing with that work, and so we'll be able to share more as time progresses.
Thank you, dear.
Just to say, Dr.
Go ahead, Bill. Go ahead, Dr. Mary.
Yeah, just to say that from our, we did some observations, I wouldn't say experiment. And what we noticed is that when you break up the nurseries in small beds, if you have a problem such as leaf spots on the leaves, you can contain it in that segment and it does not spread to the entire nursery. So one of the best practices that I would suggest that certainly we here have been practicing is to make the beds, even though you have a long bed with, it could be a concrete bed or it could be made by wooden structure. You break the beds up in small regions so that you're able to traverse it easily, and two, if you have a disease problem, it can be cauterized before it spread to the entire nursery.
And of course, it will also assist airflow. Yes.
All right. Thank you for those answers. I'm going to go back to the chat, and then I'm going to go to the hands. So this comes from Raymond Trotz. Which practice is better or best for seed nut selection from mother palm? Collection of drop nuts or harvesting dry nuts directly from the mother palm? Dr. Wallace.
I think that if you harvest directly and know exactly where that seed nut came from, that's the major advantage I would see. But if it's on the ground, you may not be sure where it came from, how long it has been there.
All right, thank you. I will now go to Reginald Andal from Grenada. Reggie, proceed with your question.
Good afternoon.
Good afternoon.
Yes. Given the slow rate of multiplication of planting material using seed nuts, is there any possibility, or have you all looked into the possibility of multiplying, let's say, your hybrids via tissue culture, maybe in collaboration with CC in Mexico?
I can start the ball rolling there and hand over to Dr. Mary. We have done two things. We have one researcher working on tissue culture in Jamaica, and we also work with CC. Up to recently, we received material from them that we would do some hardening of in Jamaica. The process is not yet perfected. It still carries its drawbacks, but yes, we are looking at that, and yes, work is being done there. Dr. Mary, could you elaborate more?
Is it in relation to the tissue culture from Mexico? Mexico is the only institution in the region that has developed the technique to such a degree as to obtain one embryo, and now I'm moving from embryo to pumule, pieces of pumule, and from the pieces of pumule to multiple plants. So you can have up to 100, up to 1,000 plantlets from one embryo. That technique is not widely dispersed around the region, for want of a better word, simply because Mexico developed the technique, and they're only going to expose us to the hardening of process which involves. So Mexico is able to supply us plantlets, and we would do the hardening off in our respective countries. I know that there are some problems with the hardening off. Mexico is willing to work with each country to make sure that those problems are resolved. Yes, we have a local program here that involves the tissue culture, but where we have reached so far is just one embryo to one plantlet. Where we want to go, and certainly for all the various countries, is to move from one embryo to many plantlets. So I think it's a very important area that we should focus some attention on. And I know the project is looking at how best we can work with Mexico to, one, to increase the population of plantlets to the various countries, and so that we can increase future production.
Thank you, Dr. Myrie. I'm going to go back to the chat before I go back to the hands. Very often, coconut trees that die from redwing as plants growing under them. Should these be used or should they be destroyed? Would these plants have the redwing nematode in their tissues? Facilitators.
Dr. Myrie, can you speak?
Dr. Myrie, that's down your street, I think. Dr.
Yes.
Red ring, as we all know, is caused by a nematode. It's red ring because when the nematode is actually transported by the vector, the American palm weaver, it has a red ring in the trunk or in the stem or in the stem or in the petiole of the leaves. If your plants, I would not recommend taking seedlings from affected red ring trees to be planted. Nematodes are actually in the phloem, or not the phloem, but in the sap of the tree. And even the seedlings can be affected by the vector in terms of how the seed going into the seedling. So I would not recommend taking seedlings from on the affected red ring trees to be planted.
Thank you, Dr. Myrie. I don't see any hands right now, so I'm going to, well, Troy, you can go ahead. Troy Augustine, your hand is up.
Yes, I was muted. I wanted to answer the question that Showin asked about selection of plants for the nursery for propagation. That will be addressed in a training that I have next week. Coming from Mexico again, we were told, or we were demonstrated, that 99% of the seeds that you collect will germinate, not 100%. You have very early germination of 33%. Then you have another 33% germination and another 33% late. To select planting material for a germplasm plot, or for selecting selecting planting material from the nursery to establish a germplasm plot, you should only select the very early 33% or the early 33% that is germinated. So all of the plants would not germinate at the same time. The first 33% that germinate, remove them, and you can put them in the bags. It's safer to do in the bags, because what I've been working with coconuts within the Ministry of Agriculture for the last 10 years. What we do, and I'm trying to get the farmers away from that, we need to stop planting coconuts, cutting off the roots, and sell it to farmers. The roots are important just the same, because the plant has to regenerate new roots to establish in the soil. So just as you buy a seedling mango or soursop or avocado in a potting bag, in a bag, we can also do coconuts that way. All of these I have to demonstrate next week, because also remember, the nematode can spread through the roots. You have a root cut, and then you have a problem. So his part of the training will be addressed. There are other issues that is preventing the what he's looking for as well, that needs to be addressed internally. But in terms of the germination collection, you can collect all the nuts you want, but you would not have 100% germination. So that I will be addressing in the training at CARDI next week, Wednesday. And we have a visit to the field to see the plot that is producing from the Mexican tissue culture coconuts. So that he can be assured, and the extension team and the farmers and the nursery persons will be there. They will be assured of everything I learned from Mexico will be highlighted, and they can be at ease.
Dr. Robin.
Yeah, go ahead, Wayne.
Just to say that we did an experiment here looking at the roots, cutting the roots and leaving the roots. And in fact, there's no difference if you cut the roots or leave the roots. Here what we do is to cut the roots and then we plant them. The thing is, when you plant them with the roots, that roots are not going to be used for feeding. That roots are going to be disintegrated, and new roots are going to be exuded from the husk, and those roots are going to be the feeder roots. And remember that when you put the seedling out in the fields, they're in the hostorium, in the nut itself. The hostorium is actually developed in such a way so that it supports the nut, that seedling, not the nut, but that seedling for X amount of time until when new roots it has come out. So there's no difference in you. Generally, what we say is to cut the when you're taking it from the nursery, we cut the roots and we do the transplant.
Any more comments from the facilitators? I just wanted to make a comment, and it just struck me before Dr. Myrie spoke, that you're having a training session next week, Troy, with CARDI, but the training material, I want to suggest that you just share it with Dr. Wallace, Dr. Myrie, and Dion Clark-Harris. They could probably add to the material you have based on their experiences. And just as Dr. Myrie is clearly indicating that he's done experiments, and the CIB has done experiments, and there's no difference between the roots when they're pruned or when they're used as is. You will get some positive feedback, I'm sure, from the experiences of these three scientists. So you could easily share your training material with them and let them see if they can add to it. I see someone making a request for Dr. Wallace's email. I want to suggest that you write to you send an email to me or the facilitators, and you will get that address from Dr. Wallace. Okay. Let me just continue going down to the chart. This looks like a comment. The Mamiku estate in St. Lucia had 400 acres of coconuts, which has been abandoned due to the bottom falling out of the market. Well, that's an experience most of the countries in the region had, and now the coconut industry is on its way back up. We have expanding markets, both for water and oil, and the bad press given to coconuts, even up till today, is always counteracted with some good press. And even recently, we circulated something in our chat group indicating the goodness of coconut, although it's a saturated oil. There's another comment in the chat group. Will the transcripts of this session be available? The session is recorded. I'm not sure if it's going to be edited before it's made available, but either Tristan or William could talk to that. My audio is very bad and I'm missing most of the discussion. So either Tristan or William could answer that in terms of making the recording available to the registered participants, right?
So just quickly, Dr. Robin, yes, this session is being recorded and it will be posted to the YouTube channel so that other persons who are on the webinar itself and those who weren't able to attend for whatever reason could also access the recording. Yeah.
Question here, if I was to rehabilitate old fields, would any financial assistance be made available? That's for Tristan and William to answer.
Right, so this sounds like something that would be requiring developmental bank assistance or some other sort of financial institution. Unfortunately, under this project, I don't think we would be able to provide financial assistance directly towards the rehabilitation of old fields. Some support will be given, of course, in terms of seed nut collection and mother palm identification at the ministerial level in order for them to really flesh out such programs. We'd also help support nursery development at both the ministerial level and some private sector levels. But in terms of being able to rehabilitate an entire field, no, we won't be able to utilize project funds towards that end. That said, of course, liaise with your relevant ministry personnel extension officers and seek the relevant guidance at the national level in terms of how best to approach this and where such resources could be drawn upon in order to further develop your own business and seeking an increase in revenue, etc.
Anybody from ITC have any comments to make on this? William, you have any comments to make on that question?
Okay. Thank you, Dr. Mary. I think we can liaise directly with Mr. James. For sure, as I validate what Tristan is saying, and for sure we can help in doing the funding proposal to reach out to development banks to rehabilitate the old plants. So we can follow up directly with Mr. James. Thank you.
Okay, thank you, William. Next question, good afternoon. Which varieties are doing best against lethal yellowing in Jamaica? In the case that a country like St. Vincent and the Grenadines comes down with lethal yellowing, what would be the approach that should be taken? Is there any country in the region that would be able to supply tolerant varieties? Dr. Myrie.
Yes, in Jamaica, the varieties that we have doing well is the Malayan dwarf, 3 color forms. Those are doing well against the disease. They are exhibiting high tolerance. We have a special Malayan dwarf that we call Special Malayan Dwarf Yellow. That is also doing very well against the disease. However, we also couple the tolerant varieties that we have with other measures, management strategies that will help us to control the disease. I know that we will be making such information available in the future to every person in the region. But just to say here now that the management technique has significantly reduced the lethal yellowing disease in the areas that it is practiced. So later on, we can go further into it. As it relates to whether or not, what's the second part of the question, Dr. Robin? What was the second part of the question?
Is there any country in the region that would be able to supply tolerant varieties?
Not in the, well, certainly we will be reluctant from here to supply seed nuts to a country that does not have the lethal alien disease. It can be done, but we would be reluctant so to do. So you're looking in the region now. In terms of outside the region, and when I say outside the region, I'm talking now about Mexico. Mexico has a very excellent variety called the Pacific Tall that they have been doing experiment with for a number of years. And that Pacific Tall is actually standing up to their lethal alien disease in Mexico. So Mexico could be a source, taking all the necessary phytosanitary problems associated with transferring seed nuts, which will be addressed later on. It is possible to get seed nuts from Mexico or from Brazil, but bearing in mind, you have to take into consideration the risk. And if you are going to import from those countries, I would strongly suggest that you have an expert to guide the process. Because remember that you also have to do some post-quarantine measures after the importation of the nuts.
Thank you, Dr. Myrie. Just a short question here. When harvesting seed nuts, how old should the palms be? I'm assuming that based on the type, there'll be a difference in age. Example, 2 to 3 or more years into production. Dr. Wallace, you want to address this?
I'm not sure there is that the age of the mother palm is a factor. I don't have that experience.
Okay. Tristan, one of
the age of the Dr. Greg,
the mother palm. The age of the mother palm.
The age of the mother palm would only be, I suppose, be important here if you are having a decrease in production, which you would normally have if the plant is getting to the end of its lifespan. But for tall variety, it is a very long lifespan. It's over 50 years. And for the
thinking about too young. I think he asked the question, maybe I get the impression if it's too soon. Is any age too soon?
Oh no, there is
right.
no such consideration.
Right.
Okay. Just some housekeeping matters there. Tristan, Dion says she cannot unmute her mic. I don't know if you all can help her from your end.
It's been resolved now, Greg.
Oh, good.
I've been trying, but I've missed my cues, so I'll just wait until the next issue.
So you finally got it unmuted now?
Yes, William has me the co-host
okay.
now.
Right. Next question. Has there been any approaches to work with APCC countries on tissue culture technology? Has there been any approaches to work with APCC countries on tissue culture technology? Can any of the facilitators give some response to this?
Greg, sorry about that. I got a little bit distracted. Could you repeat the question, please?
Has there been any approaches, obviously by us, to work with APCC countries on tissue culture technology?
I know that we have sent the Dr. Burnett, who is the person who we have working on tissue culture here, to Indonesia Sri Lanka.
Thailand.
Yes, Thailand. We sent her to Thailand, and did we send her to Sri Lanka too as well, Dr. Wallace?
I'm not sure I know about Thailand, though.
Yeah, to Thailand. And she did some work with them. So that was the only sort of collaboration we had with countries in the APCC. But yes, I'll leave it there.
All right, okay.
Under the project, we have had persons visiting countries and sharing experiences and also going to conferences. So the experiences have been brought on board through those exchange visits under this project.
Thank you, Dion. I'm going to go to Raymond Trotz. He has his hands up. Raymond, can I hear your question or comment?
Yes, I just wanted to clarify. Well, I would have liked to comment on the last question I asked also, but I will leave that for another forum. But I want to follow up on Saturdio's question on the age of the palm. Here in Guyana, in our training programs, we usually are advised, and we have arrived at an understanding consensus that it is better to harvest seed nuts from palms that are 5 years or older. The reason given is by that time we would have seen the expression of traits in a very definite way. So we are advised that I think it was not 5 years, as a matter of fact, 10 years. And many of us questioned why that long, but the reason given was that you have to allow some time to see consistently how the traits are expressed in the palm. I'm hearing now that that is not necessarily so. And I must say that I have got a palm at my little cultivation there that bears very well. It's very large. It gives an average of about over 500 mils per nut, which compares favorably with nuts coming from other parts of the country, especially Cameroon. And I started collecting from about after 1 year of bearing from the trusses. I started collecting nuts just to see for myself. But it's an important question here because we see many palms that are very good in bearing, but they might be coming into fruition after, say, the third year, fourth year, if it's a dwarf, and there's a tendency to collect from them. But we're being advised, wait until it gets to 5, 10 years, and then after that time, you can start to collect seeds. So this is an interesting revelation that is not necessarily so. You can start collecting seeds earlier. I just wanted to make that comment from Guyana. Thank you.
Any further comments from the facility?
May I please comment on that?
Yeah, go right ahead, Dr. Waller.
The only thing that relates to that, as I can see, when we're trying a variety or a hybrid, it's going to take us 12 to 14 years before we release it to farmers, because what you'll find is that the data in the first 7 years will differ from the data in the 7 years after that. But it would be assessing for suitability of planting, not genetic matters. We don't release it commercially until we see how it performs. But I don't believe that there will be a change in the genetic makeup of the progeny based on the age. But it's just that when you're assessing a particular variety, if you say, I think this will be good in Jamaica, you let it perform for a while. But I don't see it connected to the genetics. It's more like to feed adaptation.
Umaira, Belize, your hands is up. Can you proceed?
Hi, I just wanted to comment on this tissue culture part. The FCC programs to work together, Now, Mexico have the most developed methodology, but they have other initiatives around the region. What we need to do is come together and try to record these experiences, because I know in Guyana, they have some experience trying to not only do zygotic embryogenesis, but actually do mass multiplication.
Oh.
Why I bring in this also is because they have a belief that once we go into tissue culture, we will get plants faster. Nonetheless, some people feel that the nuts are the best. So if we do a program, everybody have to realize that it will take time. Just as Dr. Wallace says, it takes 14 years for release. Even for tissue culture, it will take at least 5 years when any material going in will go out that is in a good program. So anything we put in, it will take time to be released. Once it starts to release, it will be constant. And that is the advantage the Mexican have. They already did a lot of the research. They put it in, and now they in that constant where they just mass multiplying. I just want to bring that up because any research need to start now, and it will take time after we build up the varieties that we want to multiply. Also, we have to work with the small pools. It cannot be 10, 20 varieties because it will be hard to manage and the quantities will not, economically will not impact. So all these things, we have to try to bring it together and try to determine what is the best protocols for the region. If buying and multiplying, what exactly it is. I just wanted to mention that. Thanks.
Thank you, Myra. Now,
Dr. Robin, if I may.
Go ahead.
Yeah, just quickly, I know William from ITC has a feedback poll question that he would like to pose to the participants, especially as we're approaching the closure of this webinar. So I just wanted to bring that to your attention.
Okay, well, colleagues, the feedback poll is up on the screen.
And while participants are answering the poll, let me just say, and perhaps Dr. Robin, you may say this later on as well, the intention is to touch base with every country under the project at least, and have an interaction with the Ministry of Agriculture so that we could continue supporting and building out programs with relations to coconut mother palm identification and seed nut selection. So perhaps a lot of the questions may not have been answered here because I'm still seeing some more questions in the chat, and some people may have some more deep-rooted or deep-seated questions that they would want to pose, but perhaps aren't comfortable posting it here. We are going to interact directly with the countries on a one-on-one basis, ideally through the Ministries of Agriculture, the technicians and the extension officers, so that we could really get to the heart of the matter and have that type of interaction or relationship so that we could really move the industry along. So just wanted to share that with the participants as well.
While we're on this, William has sent some links in the chat. I think these links would have more detailed information on the project and aspects of the discussion this morning and more. So please record those links for your own use and future reference. Okay. Now we're going for 2 hours. And the plan was to have this webinar for 2 hours. I'm going to extend the time for 15 minutes to take some critical questions that I see in the chat. I don't see any hands up at the moment. So let me go to the chat and look at some critical questions. How long can a mother palm last before a drop of productivity?
Oh. I think the tall varieties will go further than the dwarfs. I've seen some talls go like up to 50 years. I'm not sure how far the dwarfs go because in our context, we replace them at about 15 years, but not because, or less, but not because they have dropped in productivity, but because we do hand pollination and they become too tall for our ladders. But I believe the tall varieties will go quite likely much further before they become classified as senile than the dwarfs would. Maybe 30 years for the dwarf and maybe 50 for the tall. That's just my ballpark estimation.
Thank you, Dr. Wallace. Another point for you, Dr. Wallace. Dr. Wallace, repeat an earlier mentioned point about counting leaf scars and its importance in helping to identify a tall or a dwarf tree.
Yes.
Can you elaborate a little on this, please?
Okay, the leaf scar, when a leaf falls off the palm, it leaves a scar. And if you measure the distance with 11 scars at 1.5 meter high, that's chest height and upwards, it gives you an estimate of the rate of growth of the palm. And that distinguishes the tall from the dwarf. So the higher 11 scar lengths would be indicative of the tall variety, and the smaller would be indicative of the dwarf. It's about the rate of growth, which of course distinguishes the tall variety from the short. At the same age, there would be quite a difference in height. So 11 scar would be a sample. How long? That would be 10 months. How long does it take for it to eliminate, emit 10 leaves?
Okay. I think here we end our question and answer session. And I now would ask William from the ITC to give some closing remarks and also probably talk a little bit about the follow-up action, because I indicated in discussions with Wayne and the team before, Dr. Myron Dion, that it's very important to touch base with the islands individually and have this sort of a interactive session in the field where with cameras, whether it's tablets, videos, they get information, photographs back to Dr. Wallace, Dr. Myrie, Dion, and they can interact with them looking at specific genotypes in the field and discussing all what we discussed today about selections. So I'm going to hand over to William now to give some, from the ITC, to give some closing remarks. William.
Thank you, Dr. Robin. So I think just from the ITC point of view, we'd just like to say thank you very much. Thank you, Dr. Wallace, Dr. Myrie, CARDI, Dr. Robin for your moderation. And through you, Dr. Wallace and Dr. Myrie, please extend our gratitude to the CIB, to the board. You have always been willing to support us, to support the project. The CIB is doing magnificent work for the region as well, so we'd like to thank you again. CARDI, of course, thank you for facilitating this workshop and reaching out to the different stakeholders, to the different ministries. in terms of the next steps, as you all know, we work with the Alliances for Action approach, which is basically inclusiveness and participatory approach for the region. The idea after this is taking all this knowledge that we have been shared by Dr. Wallace, Dr. Mary, Dr. Clark Harris as well from CARDI. The idea is to reach out to the different ministries of agriculture and also the private sectors and farmer cooperatives in the different countries of the project. And then let's do the same training, but in the field. Of course, the first thing is to take care of our health, taking care of the COVID. But the idea is now to plan field activities, to go out with the farmers itself, to go out with the extension officers of the ministry, to select these high quality modern palm and seed nuts in the field. And the idea, of course, is to use this mother palm to establish or strengthen ministries at the sites of the Ministry of Agriculture and the site of CARDI. The idea is for the smallholder farmers in particular, to be able to have access to high quality planting material. As you might know, production in the Caribbean and worldwide of coconuts is stagnant. So we need to promote this production, and then our role is also to find the markets for those production. How do we add value to the coconut that we produce? How do we produce high value added products, coconut oil, coconut soap, now for the COVID times, coconut water, and then going to more innovative products like coconut sugar, for example. How do we add value to that as well? So just from the International Trade Center, Alliances for Action team, and CARDI, we'd like to extend our gratitude to our partners, and in particular to our donors, of course, the European Union, the CARI Forum Directorate, the Office for Africa, Caribbean, and the Pacific Secretariat. Thanks a million. I have, as shared by Dr. Robin, I have put in the chat our social media accounts for Alliances for Action, for CARDI, the Facebook group, which we encourage you to go in the Facebook group and ask your questions, the same one that you're doing in the chat. So, and subscribe to the group so many people can address your questions, and then this could be a platform to discuss within the region and also with other coconut producing economies. So thanks a million. We look forward to continue working together with you. And I know it's reaching lunchtime for the Caribbean, so just to end up, bon appetit to all of you, and thanks again. I hand back to you, Dr. Robin.
Thank you, William. I should also say there are a lot of compliments in the chat, congratulating us on a very informative webinar. There's one thing that strikes me is that we need to form a sort of a CARDI, CIB, CCS, a sort of a group that all these islands that need on-hand information readily can just send in an email and they get a response. Not wait for the project action and so, because I sense the interactions we have with Grenada, I sense that they're going into a training, and I sense we can make a ready intervention with the experience we have, the project and its collaborators in that training session. We can add to it, I'm sure. So something is coming out of this in addition to everything else. I think we should have this nexus. William, Ben from ITC as the front people, Wayne, Dr. Myrie, Dr. Wallace, Dion Clark Harris, Kristen, that people could just send in an email like a group chat and somebody will respond to them immediately on the issues they want to address. With those comments, I want to close the webinar. I enjoyed doing it, and all the best for the rest of the day. Take care and stay safe. Thank you.
Thank you for having me. Goodbye, everyone.
It's always a pleasure, Dr. Wallace.
Thank you very much.
Same here.
Goodbye, everyone.
All right.
Bye, everyone.
Bye.
Bye.